the topic of gram staining in the subject of microbiology, which is a very basic and important topic to understand. So let's start our video. So what do you understand by gram staining? Before that, we will look into our topics that we will cover today. So first of all, we will cover history. Then we will see the difference between gram positive and gram negative bacterial cell wall. Then the procedure of gram staining, and then the modification and interpretation of gram staining. So the history of gram staining. The gram stain was devised by the Danish physician Hans Christian Gram while working in Berlin in 1883, and he later published this procedure in 1884. He was the scientist, as you are seeing in the pic. Now the differences between the cell wall of gram positive bacteria and gram negative bacteria. As you can see, the main characteristic difference between both of them are the first one is peptidoglycan layer, which is very thick in gram positive and very thin in gram negative. You can see in the pic also, the peptidoglycan layer is very thick in gram positive. Let me take the highlighter. Now at the third position of the tetrapeptide side chain, in gram positive L-lysine is present, and in gram negative mesodiaminopamelic acid is present. Then the pentaglycine bridge is present in gram positive and absent in gram negative. The lipid content of gram positive is nil or scanty, while the gram negative has more of lipid content. The lipopolysaccharide layer is absent in gram positive and present in gram negative, which plays the role of endotoxin. The teichoic acid is present in gram positive and absent in gram negative. The variety of amino acids are very few in gram positive, and there are several types of amino acids in gram negative. The aromatic amino acids are absent in gram positive and present in gram negative. So this is the pic showing gram positive and gram negative. So in gram negative, we have the lipopolysaccharide layer, which has O specific side chains, core polysaccharide, lipid A part. This all comprises the lipopolysaccharide. This is the outer membrane of the cell wall. This is the periplasmic space and the few less amount of peptidoglycan, and this is the plasma membrane. There is presence of outer membrane proteins also, and there is one specific protein known as bronze lipoprotein in the gram negative. In the gram positive, it is very simple as compared to gram negative. There is presence of lipoteichoic acid and teichoic acid in the peptidoglycan layer, and this is the periplasmic space, and this is the plasma membrane. So the procedure of gram staining is, first one, we have to do fixation. The fixation is done by the smear made on the slide from the bacterial culture, or the specimen is air dried and then heat fixed. The step first is the primary stain. The primary stain is stained with paracyanilin dyes such as crystal violet or Genesian violet or methyl violet for 1 minute. Then the slide is rinsed with water. The crystal violet stains all the bacteria violet in color, irrespective of whether they are gram positive or gram negative. The second step is mordant. The mordant is played by the grams iodine, and then it is poured over the slide for 1 minute, and then the slide is rinsed with water. Grams iodine acts as a mordant, binds to the dye to form bigger diiodine complexes in the cytoplasm. Step 3 is decolorization. The decolorization is pouring of few drops of decolorizer to the smear, example acetone, for 1 to 2 seconds, or ethyl alcohol for 20 to 30 seconds, or we can use acetone alcohol for 10 seconds. Most commonly we use the acetone. The slide is immediately rinsed with water. Decolorizer the remove the primary stain from gram negative bacteria, while the gram positive bacteria retain the primary stain. The fourth is counter stain. The counter stain can be secondary stain such as safranin or dilute carbol fusion is added for 30 seconds. It imparts the pink color or red color to the gram negative bacteria. Alternatively, neutral red may also be used as counter stain, especially for gonococci. The slide is rinsed in tap water, dried, and then examined under oil immersion objective. So here in this diagram, it is showing the primary stain, crystal violet for 1 minute, then rinse it with water. Then step 2 is mordant, grams iodine for 1 minute, then rinse. Then step 3 is decolorization, acetone for 2 to 3 seconds, and then rinse immediately. Step 4 is counter stain, dilute carbol fusion or safranin for 30 seconds, and then water and rinse. So the interpretation of gram stain. The interpretation of gram stain can be done by gram positive and gram negative. The gram positive resist decolorization and retain the color of primary stain, that is violet, and the gram negative bacteria decolorize and therefore take the counter stain appear pink. So these are the examples of gram negative and gram positive bacteria. The gram positive bacteria can be lactic acid that is present in the yogurt, bifidobacterium, staph aureus, mycobacterium tuberculosis, anthrax, enterococci, streptococcus pneumoniae, hemolytic bacteria, and clostridium botulinum. The gram-negative bacteria can be acetic acid bacteria, Xanthomonas, Zymonas, Pentuia, E. coli, Vibrio, Salmonella, Pseudomonas, and periodontal disease bacteria. The modification of gram staining are of three types. First is Kopelov and Biermann's modification. Here the primary stain and the counter stain are methyl violet and basic fusion respectively. The second modification is Jansen's modification. In this method, it involves the use of absolute alcohol as decolorizers and neutral red as counter stain. It is useful for meningococci and gonococci. The third one is Brown and Bren modification, and this is used for actinomycetes. So this is the topic of gram staining, guys. I hope you understand the topic and very well, and you understand the procedure also. So that's all for today, and I hope we will meet in the next video. And up till then, please like, share, and subscribe my channel, and also please press the bell icon for upcoming videos. Thank you, guys.